or some results, whatever you want, from memory. That'd be all right. Now it says on the handout, bacteria and yeast and milk early on. Now if it says early on, that means what predominates for living things when you first buy the milk at the store? That'd be Streptococcus and Lactobacillus. The main ones that are there when you first buy the milk or early on. Then later on means what predominates for living things when milk's gone really, really sour and really, really bad. That'd be Pseudomonas and yeast. It also says why sour. One particular bacterium makes it go sour in some cases. It's called Lactobacillus. I want to connect those terms, sour and Lactobacillus. Now it says bacteria on raw chicken. These two bacteria, you should know, are on the surface of most raw chicken you buy at the store. They're called Salmonella and Campylobacter. Salmonella and Campylobacter. We are not going to have any gram stain slides to look at, but the ones listed there, I could ask you their gram stain result in words. It would be in words. So Streptococcus is a gram-positive coccus in chains. Strep means chain. Lactobacillus is a gram-positive rod in chains without endospores. Without. Now the last three, Pseudomonas, Campylobacter, Salmonella, are the same thing. They're a gram-negative rod, individual cells. Little pink rods. All three of those are a gram-negative rod, individual cells. So I may ask you some of those in words. Now find your notes on Lab 6 and the scientific method. Those 5 steps we use for our experiments. Know the highlights of the 5 steps. Step 1, you accumulate previous data. You put down information about your topic. Step 2, you formulate your hypothesis. This is an educated guess or prediction based on the previous data. Once you put down your hypothesis, you should stop the steps for a while and define certain terms for your experiment. Beginning with the independent variable. This is the variable whose effect is being measured. The dependent variable might be affected by the independent variable. The control group and the experimental group receive the exact same conditions, except the control group does not receive independent variable. The experimental group does receive independent variable. I want y'all to notice that I left off the practical, the controlled variables left off. However, the handout does say variables and groups given sample hypothesis. So if I give you some hypothesis next time, can you pick out those 4 things? Independent, dependent, control, experimental. Let's try for practice again the one in your notes about saccharin. We said one day that ingesting saccharin will cause an increase in bladder cancer rates. For that one, what's the independent variable? Saccharin, measuring the effect of saccharin. The dependent variable is the bladder cancer rates. The control group will be a bunch of lab rats that receive no saccharin in their diet at all. The experimental group will be an equal number of lab rats that receive saccharin every day in their diet. Now, I think your notes is about saccharin. I believe your write-up was about smokeless tobacco. Does that sound right? On the practical, it'll be something different. It'll be a new one. It'll be similar to those, not the same thing. Can you pick out those 4 things for my hypothesis? Now, once you put down those things for your experiment, then do step 3. Conduct the experiment. Step 4, report your results. Step 5, draw conclusions. All we do in step 5 is relate the results back to the hypothesis. Do the results support the hypothesis or not? Now the left-hand column says list of disinfectants in B3. Now I'm talking about page 9 of your lab manual, page 9. For B3 there, that list wants you to know how are they used and are they antiseptic? Are they safe on living tissue? So you should know that these days phenol is used in throat lozenges. Antiseptic, yes. Hexachlorophene gets added sometimes to soap and detergent. Antiseptic, yes. Cresol is the active ingredient of Lysol. Antiseptic, no. Alcohol is the active ingredient of Scope and Listerine. It's also used on skin. Antiseptic, yes. Chlorine is the active ingredient of bleach. It's also found in swimming pools and drinking water. Antiseptic, I'd say yes, in very, very, very small doses. Iodine kills bacteria excellent on the skin. Antiseptic, yes. Hydrogen peroxide is used on cuts sometimes. Antiseptic, yes. Now take a look at the handout in the right-hand column. It says diagram Lysol bleach. Now these are on the back of the review sheet, at the back. These are some averages we've got in this class for a few years. Now it says Bacillus subtilis. The idea would be to streak out Bacillus subtilis with a cotton swab and place a paper disc on there that is saturated with Lysol or Listerine or whatever. Then check for a clear zone. The bigger the clear zone is, the better it kills. Now on your practical, once you recognize these as the expected results. Now you can memorize the numbers if you like. My advice is to focus on concepts. What concepts make it look like that on the results? For example, Bacillus subtilis has endospores. If some disinfectant is to kill endospores, it must kill excellent. Sure enough, Lysol and bleach kill excellent. So pretty good clear zones there, despite the endospores. Listerine kills fair and does not kill endospores. Scope kills good and does not kill endospores. On the other hand, Staphylococcus epidermidis does not make endospores. So some bigger clear zones there, except Listerine. But in general, bigger clear zones. You may see a question like this next time. It may say Bacillus subtilis, Lysol, 24 millimeters. In that case, the answer is the results are expected. That's what's on here, as expected. But I may say this, Bacillus subtilis, Lysol, 0 millimeters. The answer is the results are not expected. So something very, very different than what's on there would be not expected. Now it says on the handout there, flaming endospores. Now the concept is, if you flame for 1 second, that should not kill all the endospores. So it says there, control is positive for growth, which it should be, he didn't flame that. 1 second of flaming also has growth. That's expected. If you see that right there, that'd be expected. 1 second didn't kill them all, not supposed to. Now kind of make a note to yourself that I may do this. It may say control, positive for growth. 1 second of flaming, negative for growth. That'd be not expected as an answer. Results are not expected. If 1 second kills them all, negative growth, that's not expected. If you flame for 5 full seconds, that should kill everything, including all endospores. So control, positive for growth, and 5 seconds of flaming, negative for growth, that's expected. That should happen. Again, the card next time may say this. It may say control positive, and 5 seconds of flaming also positive for growth. That'd be not expected. If 5 seconds of flaming didn't kill them all, they're both positive there, that's not expected as an answer. Now it says boiling water. Bacillus subtilis has endospores that survive pretty well. So after being boiled, it averages about 80 colonies. E. coli, after being boiled, averages 0 colonies. I give you those two numbers that be expected. Endospores is key. Now sometimes I tend to reverse those numbers, and that'd be not expected. Staphylococcus epidermidis hangs out in clusters. Because of what I call the cluster effect, it survives salt really well. E. coli, as individual cells, gets surrounded by salt and loses water so much, it doesn't survive salt too well. So again, those two colony counts, I'll give you those two, it'd be expected. If I reverse those two, it'd be not expected. Now take a look at the front of the review sheet in Lab 7. Find your notes from Lab 7 on a man named Ehrlich. He tried to find, a long time ago, a cure for syphilis. His medication became known as Salvarsan. I'd call Salvarsan the first successful antimicrobial drug. It had some success anyway, treating syphilis. So connect those terms there, Ehrlich, syphilis, and Salvarsan. I sometimes ask this question: what was Fleming trying to grow and study? Staphylococcus aureus. He was trying to grow that one, study it. But what did Fleming grow accidentally as a contamination? Penicillium notatum was an accident. He discovered then, by accident, penicillin. A few years later, a man named Florey separated the penicillin from the mold that makes it. Florey came up with pure penicillin with no mold. I sometimes ask this date. In 1941, they began to use penicillin on patients. The first antibiotic ever used. Now find your notes on the 4 classes of antibiotics. This is based on how they kill. The first class is called cell wall synthesis. They kill by preventing or blocking cell wall synthesis. You may want to jot down from memory on the back of the answer sheet next time, APBB. Ampicillin, penicillin, bacitracin, and vancomycin. Those are all in a class called cell wall synthesis. You may see a question like this next time. In what class is ampicillin? That could be a question. That'd be cell wall synthesis. Also know from the experiment that penicillin is not broad spectrum. It kills quite well the gram-positive bacteria. It does not kill well gram-negatives. Be sure to connect these two terms, penicillin and peptidoglycan. It messes up that part of the cell wall, which is going to be a lot to mess up if you're gram-positive. The second class is called protein synthesis. They kill by preventing or blocking protein synthesis. The examples to those fill the word get. So gentamicin, erythromycin, and tetracycline are in that class. I also want you to know how they do this. They get inside bacteria and they bind to the ribosomes, make them non-functional. Bacteria cannot make protein, will die. Also know from the experiment that erythromycin is not broad spectrum. It kills gram-positive bacteria well. It does not kill gram-negatives well. And the reason is erythromycin is not lipid soluble. Is not. It cannot dissolve very well through the gram-negative cell wall, which is mainly lipid. On the other hand, tetracycline is broad spectrum. It kills gram-positives well. It also kills gram-negatives well. Because tetracycline is lipid soluble. The third class is called the plasma membrane class. Plasma membrane. They disrupt, they mess up the bacterium's plasma membrane. The examples are PMZ, polymyxin, nystatin, and Zybox. The last class is called nucleic acid synthesis. They kill by preventing or blocking nucleic acid synthesis. The bacterium cannot make DNA or RNA or both, they will die. If you jot down DNA and RNA and circle the middle two letters, that's the example. Actinomycin and rifampin are examples. Now find your notes on the Kirby-Bauer test. Kirby-Bauer. We're trying to find out here which antibiotic is going to work best for our patient. This utilizes something called a McFarland tube. This is a standard concentration of bacteria used in the Kirby-Bauer test. I connect McFarland and bacteria. Once you achieve that concentration with your patient tube, then do a double streak with a cotton swab. Then place your paper disc on there, perhaps penicillin. As usual, the bigger the clear zone is, the better it kills. To make a decision how to treat the patient, it's going to be a chart. Ours is on page 11. Page 11. On the chart on page 11, do not, of course, memorize the numbers, but do memorize the final conclusion for each category. You should know that R is resistant. The conclusion is don't use it. Doesn't work. S, susceptible. The conclusion is use it. Works great. I stands for indeterminate. The conclusion is we need more testing. MS stands for moderately susceptible. The conclusion is it will help the patient if you can use higher doses than normal of the antibiotic safely, or concentrate the antibiotic in the body region of the infection. So for each one, know the final conclusion. Now the handout says in the right-hand column, the Kirby-Bauer results in the right-hand column. So I'm going to do it this way. On some table next time, there'll be a copy of this chart sitting there on the table to look at. The question might say this: if you test penicillin and your clear zone is 30 millimeters, what's your conclusion? Well, the answer would be use it. Works great. That's S. So can you find the right category and the conclusion to know for that category? Let's try one more. If you try erythromycin, your clear zone is 16 millimeters. Conclusion? We need more testing. That's I category. So can you find the right category there and get the conclusion? So again, I'll give you all the chart, and I'll tell you which one we're testing and the clear zone diameter. You got to find the category and know the conclusion for that category. Now the right-hand column says diagram Kirby-Bauer results. These are on the back of the review sheet again, on the back. These are averages over a number of years in this class. You can memorize the numbers if you like. Again, my advice is think about concepts here. For example, E. coli is gram-negative. Penicillin and erythromycin don't kill those very well. So if we say 9 and 11, those are barely bigger than a disc. They're small, they're R. Don't work too well. Tetracycline, being broad spectrum, kills gram-negative as well, big clear zone. Now, Staph epidermidis is gram-positive. All three of those kill really well gram-positive, big clear zones. Now, if you tried penicillin against Staph in our class, you may have gotten about 14. That's a strange case there, I think, a mutation, I think. We're going to go with the typical result, which is 55 millimeters in that test, huge clear zone. Now, once again, you may see a question like this next time. It may say E. coli, penicillin, 55 millimeters. Conclusion? The results are not expected would be your answer. If I say E. coli, penicillin, 9 millimeters, you choose results are expected. So the same thing as that, expected. Very, very different than that would be not expected. Now the back to the front of the review sheet, and find your notes on the so-called MIC test, MIC. And you may want to go ahead and turn to page 12 of your lab manuals. You may have some diagrams on page 12. Now we talked about setting up 9 test tubes. Incubate and then draw a conclusion next day. The handout says this. What's the purpose of tubes 1 through 7? Well, to test one antibiotic at 7 different concentrations. In the first 7 tubes, we test one antibiotic, perhaps penicillin, at 7 different concentrations. Now the purpose of test tube 8. Make sure the patient bacteria were alive to begin with in that culture. The purpose of test tube 9, to make sure the antibiotic solution was sterile and not contaminated. It says, what are the contents of test tubes 1 through 7? You would put 3 things in those first 7 tubes. You put nutrient broth, patient bacteria, and the antibiotic solution. In test tube 8, you put just 2 things, nutrient broth and patient bacteria. In order for results to be called valid, test tube 8 must come back cloudy. That means, yes, the patient bacteria were alive to begin with. In test tube 9, we put 2 things, nutrient broth and the antibiotic solution. In order for our results to be called valid, test tube 9 must come back clear. That means, yes, the antibiotic solution was sterile. Now, we're going to skip the dose for a moment, but look at the right-hand column. It says diagram MIC results. I will draw these on a card. I'm going to draw 1 row of tubes on a card with my squiggly line being cloudy. Ask for a conclusion. On that kind of question, what do you check first? The controls, 8 and 9, to avoid confusion. Now, for example, if test tube 8 is cloudy and test tube 9 is clear, what does that mean? The results are valid. They're valid. Then what do you check for? The lowest concentration that made it clear and killed it. That's called MIC. Listen up. On your notes, MIC was what? 8? On the practical, it could be 8, or 4, or 16, or whatever. It's the lowest concentration across there that made it clear and did kill them. That's called MIC. Now, our blood will tend to dilute things. So the dose we're going to use for our purposes is MIC times 3. So MIC is 8, dose is 24, for example. Now, what's going to be the answer next time if test tube 8 is clear? Start over. The patient bacteria were dead to begin with. Nothing else matters. If test tube 8 is clear, it must start over. The patient bacteria were dead to begin with. What's the answer next time if they're all cloudy except 9's clear? The results are valid, the bacterium must be resistant. If they're all cloudy except 9's clear, the results are valid, the bacterium is resistant. What's the answer next time if all 9 tubes are cloudy? Start over. Start over. The antibiotic solution was contaminated. If they're all cloudy, which means 9's cloudy, you must start over. The antibiotic solution was contaminated. So on a card, I'm going to draw 1 row of tubes with squiggly lines and ask for a conclusion. Now find your notes on Lab 8. In Lab 8, we had a diagram one day of the components of our blood. Our blood is 55% plasma, liquid part. It is 45% formed elements, the solid part. Our plasma is 90% water and 10% dissolved substances. The so-called formed elements are red blood cells, white blood cells, and platelets. Together, they make up 45% of our blood. The function of platelets is to form the initial blood clot. We compared red blood cells, or erythrocytes, versus white blood cells, leukocytes. Usually a white blood cell is 2 or 3 times bigger than a red blood cell. The primary function of a red blood cell is to transport oxygen from our lungs out to our cells. Our cells must have oxygen for a step called electron transport. The secondary function of a red blood cell is to transport CO2, carbon dioxide, from our cells to our lungs to get rid of it. Our cells make so much CO2 in a step called Krebs cycle. The general function of a white blood cell is to fight infection. A red blood cell has no nucleus and no DNA. Don't worry about why that is, just don't have those things. However, a white blood cell has a nucleus and DNA. Now find your equation on hemoglobin. Hemoglobin, or Hb, plus 4 O2s gives a complex of hemoglobin and oxygen, HbO2/4. Now that takes place all the time, from left to right in our lungs. We are creating what you might call oxygen-rich blood, which winds up taken out to our cells by arteries. Let's again be careful when we say this. Arteries always take blood away from the heart. It is almost always oxygen-rich. Now that equation, the equation now takes place from right to left all over the body. The O2 comes off and goes to our cells. So right to left takes place all the time, all over the body. Getting the O2 out to our cells. We are creating, though, oxygen-poor blood, which gets picked up by our veins. So here we go. Veins always take blood towards the heart. It's almost always oxygen-poor. Now we'll try some numbers there. Each molecule of hemoglobin can carry 4 O2s at a time. Each of our tiny red blood cells contains about 200 million molecules of hemoglobin. If we multiply those together, each of our tiny red blood cells can carry up to 800 million molecules of O2 at the same time. Now the total white count. This is the total number of white blood cells for some patient in a cubic millimeter of blood. The normal range is 5,000 to 10,000. Now, what I tend to do right here is give you all a patient's total white count. I give you a number. Ask for a conclusion. Let's try for practice the ones on page 13. On page 13, this is B3. The first white count is 20,100. That's too high and probably infection. 2,200 is too low, which may be most likely medications or a need for blood thinners, or the patient might have AIDS. 101,400 is bizarre too high and leukemia, almost certainly. 7,500 is within normal range. On your practical, not the exact same numbers, to be similar, and you pick out the right conclusion. Now find your chart on Never Let Monkeys Eat Bananas, 5 types. Now, all the chart is going to be fair game for a question. You got to know the percentages, you got to know functions, and the so-called if elevated. And be sure to make a note to yourself, on the lab practical, the chart goes with the slides. The slides. For example, if you see this cell right here next time, the question might say this, what is it? The eosinophil. Or it might say this, what's the functions of that white blood cell type in the chart? What's the so-called normal percentages? So the chart goes with, on the questions, these slides, slide stations. Now it says the differential white count. This gives us the 5 kinds of white blood cells and their percentages for the patient. A doctor compares the patient's percentages to normal. If one's too high for a patient, that could mean something. So the right-hand column says the diagram differential white counts. Let's try these for practice on page 13 again. This is B6. Each column going down the page is a patient. Now, if one of these is too high, the rest pretty much have to be, in some cases, too low. Only 100 points to go around there. Not too concerned at all about what's too low. The question is what's too high. For example, patient 1 may have an allergic reaction or a parasite infection because eosinophils, 37%, is too high. Patient 2 may have leukemia. Basophils, 24%, too high. Patient 3 may have a viral infection. Lymphocytes, 45%, too high. Patient 4 is within normal range. Patient 5 may have a bacterial infection. Neutrophils, 84%, too high. Patient 6 may have listeriosis because monocytes, 37%, that's too high. Again, on your practical, not be the exact same numbers. It'll be similar. Again, don't worry about for your practical what's too low. Which one's too high? What's that mean? Now the review sheet mentions the slides over here. Now, I'm going to turn these lights on in a minute. Take a look at these. There should be a white blood cell at the tip of the pointer that matches the card. If you can't see it good enough, then try the find knob. If I still can't see it good enough or it got moved, then find me. I'll fix the slide. I'll be in here or my office. Okay, that's all.
Yeah, buddy.